The latent stage of Mycobacterium tuberculosis (MTB) infection is defined by the switch from an active to a non-replicating condition, caused by the formation and consequent necrosis of the granuloma in the lungs. In the granuloma, MTB will be exposed to various conditions such as hypoxia, increased nitric oxide and carbon oxide levels, low pH, and limited nutrients that will challenge and induce the expression of different genes that cause metabolic reprogramming. In the bacteria, there are two sensor kinases, DOS-S and DOS-T, which work together to activate the transcriptional response regulator DOS-R. During hypoxia, DOS-T is the first to respond as it is more sensitive to oxygen changes than DOS-S and induces the DOS-R regulon with DOS-S. As the oxygen becomes more and more limited, DOS-T loses its functionality and DOS-S continues to maintain the induction of genes alone. Both the sensor kinases are very similar, sharing 62.5% similarities in amino acids and both containing GAF-A and GAF-B tandem domains in the N-terminal. The GAF domains of the two sensory kinases are important to detect the different environmental conditions and regulate the kinase activities of DOS-S and DOS-T. Indeed, the kinase activity of these two proteins is regulated by the oxidation and binding state of the heme ion in the GAF domain. Therefore, the binding of nitric oxide or carbon oxide initiates the phosphoryl transfer to DOS-R. Once DOS-R is activated, it works with DOS-S, ultimately regulating a large regulon with 48 genes, the DOS-R regulon. The DOS-R regulon is the key to the survival of MTB during latency. Despite the lack of oxygen, the DOS-R regulon allows the bacteria to survive under anaerobic conditions, upkeeping ATP levels, redox states, and various metabolic processes. Due to its adaptation to surviving in these conditions, Mycobacterium tuberculosis can survive dormant in the host body for months or even years. The transcription of the DOS-R regulon is induced by the DOS-R, which binds to the DNA and allows for transcription factors recruitment. Although MTB is an aerobic bacterium, it is able to reprogram its metabolism into surviving hypoxic conditions. This is thought to be done by some of the genes regulated by the DOS-R regulon, such as NARX and NARC2. Evidence suggests that the NARC2 is a nitrate-nitrite transporter. The protein is situated in the cell membrane as suggested by the many alpha helixes that make up the protein. NARC2 allows for the flow of nitrate into the cell, which is then used as an electron acceptor for the bacteria metabolism instead of oxygen. The bacteria will remain in the latent stage until reactivation occurs.